medicine paradigm. First, of course, is cancer therapy. We are working very intensively on cancer therapy and blood-brain barrier research. The blood-brain barrier research is also very versatile. I have a strong collaboration from my previous postdoc. I was doing a lot of blood-brain barrier studies and experiments under the supervision of Professor Foster at Würzburg University Hospital. And since then, we are very closely connected. We are generating the new cell lines for the blood-brain barrier. We are developing new assays, in vitro, in vivo assays, how to monitor the different substances flowing up and down through the blood-brain barrier. And we are very successful at this. And now we are also very interested to implement medical informatics, biomedical informatics in this research, to do some models using different techniques, and we are quite successful. So we are also doing some cardiovascular disease research. My collaborators at the University of Würzburg, but this is just a minor project, and this is not really the focus of our laboratory. And we're also doing a lot of experiments in terms of the infectious diseases in immunology, because my background in infectious diseases, I was serving as a doctor in the hospital, in the infectious diseases and immunology hospital, treating some patients with HIV, with hepatitis, diarrhea, and flu, influenza, and various other infectious diseases. So since then, I was involved very heavily in infectious diseases research, and also my supervisor, my former supervisor, Professor Dandika, who's the head of biomedical informatics and bioinformatics research at the University of Würzburg. He has a very strong connections with parasitologists and people doing infectious diseases research. Therefore, every year we are gathering together and doing like training for the master students, bachelor students, PhD students, kind of summer school every year on a regular basis. We are calling it medical informatics, bioinformatics for infectious diseases course, where we are talking about the strategies, about the new strategies, AI in the bioinformatics, and how we can implement them for the infectious diseases. And just to start with, I would like to introduce the first project. It's called Robotic Anti-Cancer Therapy. So what is it all about? This was already published a couple of years ago, and since then we are very heavily involved in the continuation of the project, together with my colleagues from the hospital, with Professor Prof and with some other oncologists, and also with some mathematicians. Professor from the US, Nosonovsky, he is the full professor at the University of Wisconsin. He is also serving as a full professor at ITMO University, and he is heavily involved in the mathematical kind of backend, so to say, backend to integrate all his knowledge in mathematics to provide the mathematical modeling for biomedical research as well. But we are working together. So what is all about? What is the robotic therapy? I would like to say a couple of words. And this is just to advertise our new paradigm. We are still working on it, so it's more like hypothesis, so you have to take it with a grain of salt, because all I'm just talking about is more like theory. We have to prove it in vivo, in vitro, we're still doing in vitro, but in vivo in a clinical setting. So this is kind of an early phase of the research. For that, we are working on the concept we are calling chromatopsis. And what is the chromatopsis? It's shown here. When you have a normal chromosome, you have some kind of disintegration of the chromosome, like fragmentation of the chromosomes, and then you have the assembly of these fragments. And after that, you have some so-called altered chromosome, which is partially functional, partially not. Some genes are impaired, some genes are overexpressed, so it's more like chaotic. So mechanism, the detailed mechanism for this process is not really very well understood. There is a lot of research going on, and we are right now working with a lot of researchers from abroad, like Professor in the US, Professor Tan in the US, and we are trying to investigate this particular mechanism. But we are also interested in how we can implement this particular mechanism if we have a cancer progression, because if you have a cancer progression, then you have like a cancer stem cells that's going to divide, and if you have this particular mechanism in a cancer cell, what's going to happen? And to answer these questions, we try to replicate this particular mechanism in a cell culture, and we have found that there are some kind of fingerprints, and these fingerprints are well known to describe the genomic instability. So when you got like micronuclear bridges formation and some elements which are associated with the chromatopsis, they are usually harmful for the cell, and the cell just going to die. And the pathologists, they will see these fingerprints and biomarkers in the cancer cells. So we actually developed this particular assay, in vitro assay, and Professor Prof is working on this particular project for many years, like 15 years already. He had published a lot of papers on this particular biomarkers to describe the genomic chaos. And we have some cells, some cancer cells, like glioblastoma. They usually express in the brain, medulloblastoma, glioblastoma. Some cells are going from the epithelial cells and also the muscle cells, rhabdomyosarcoma, because this study was done on rhabdomyosarcoma, and where we try to investigate this particular biomarkers in cancer cells, because not all cancer cells, they have this particular mechanism of, so to say, development, yeah? And only 15% of cancer, so some cancer are kind of robust, and they don't really have this particular, but we can actually induce this mechanism to lead the cancer to the extinction. So this is the idea of this particular mechanism. We are calling it body anti-cancer therapy, which is based specifically on this chromatopsis. There are different variants: chromatopsis, chromoplexus, chromorexis, and so on. The whole paradigm is called chromoanagenesis. And we are just talking about the chromatopsis, but of course it's much more complicated than that, yeah? And the project was to investigate and to model the situation, using some proteins which are very important for the cancer survival. And some of the proteins like BCL2, they are known that they're very important because they either protect the cells from the apoptotic event, or they can stimulate the cells, and the cell will undergo through the apoptosis and will die. So this is the idea. And of course, BCL2 is very well known, like anti-apoptotic protein, which is here. And we were thinking about what is going to happen if we have a chromatopsis in the cancer cell, and how it will actually correlate with mutation, because if you have some kind of chromosomal rearrangement, which is exactly what the chromatopsis is, you have also the accumulation of different mutations in a cancer cells. For instance, in reality, you have like 10 and minus 10 power mutations per nucleotide per cell division, which is of course drastically will be increased if the cell will be cancerous, for instance, 1,000 times, yeah? And of course the 1,000 times more if you have a chromatoptic event. So you will actually generate a lot of mutations. So to investigate this, we just developed the pipeline, and we took the sequence of the BCL2 gene and replicated the mutation rate for this particular protein using some Monte Carlo algorithms. And we, stochastic algorithms, we actually mutated the gene, and from gene we came up to the protein, and we tried to investigate the stability and the function of the protein and how it actually correlates with the mutational rate if we have a chromatoptic event. And for that, we used 10 and minus 5 power divisions per cell, per nucleotide division, and we saw that the stability of the cell is impaired. So once you get the mutation of the protein, you are decreasing the stability. For instance, this is Monte Carlo of the control and the mutated protein. So BCL2 mutated and the control. And here you can see that the function, the energy function is going up. It's going up here, which means that the protein becomes thermodynamically less stable than the control. And if you look at some kind of, it's not really the free energy landscape, but this triangle is a kind of 2D approximation of the protein folding. So it loses the triangle shape here, which means that protein becomes kind of unstable, and the folding is also a kind of impaired. Of course, it's not really a folding, because for folding you need the final kind of thing. Yeah. In the dynamics, we're usually doing this folding, molecular folding, protein folding, and different metastates, and here's just kind of approximation. And after that, we try to investigate the protein-protein interaction, because it is known that the cascade for the apoptotic cascade is Buck BCL2 Buck pathway. And we try to investigate how the mutated BCL2 interacts with the Buck proteins. And we saw that also this interaction is impaired. So the energy, the affinity is decreasing. Therefore, for this particular study, we concluded that if we have a chromatoptic event in the cancer cell from the experiment, and a particular mutational rate per cell, per division per nucleotide, we can actually correlate this, what will happen with some key proteins which protect the cancer cell against the apoptosis. And this is also nicely explained in the experiment. As you can see here, we have the frequency of the cells with micronuclei, and once you get, but this is directly calculated from the experiment, so this is not a prediction. And once you get more cloning stages, you will have the accumulation of this particular biomarkers here. And then you can also calculate the ergodic, so to say, breakage. The ergodic breakage is very important because this is a kind of a threshold. After that, you can actually induce the degradation of the cell, the cancer cell. And this is about kind of 15%. After that, when you have a chromatoptic, this event close to 15%, 10-15%, then you can actually stimulate the cell to degrade. And right now we are searching for different protein target, for different molecules, drug-like molecules, where we can actually use this paradigm to stimulate the cells using the in vitro